What's number one? Basically the orange portion that you're seeing. Oh, thank you so much. What's the orange portion that you're seeing?
Brain.
Well, the whole thing's the brain, but it's a specific part of the brain, right?
Cerebrum.
It's called the cerebrum, right? So the orange part that you're seeing there is the whole thing. You're right. The whole thing, brain. But this part, that orange part that you're seeing, number one, you guys see the little number one up there? That's going to be specifically the cerebrum, okay? Cerebrum.
Yellow, you mean?
A peach. Let's call it peach, right? Peach, cream, I don't know whatever color that is. This thing right here, all this that you see here, that, I'm going to call it yellow. That's going to be the cerebrum, right? Okay. What's the significance of the cerebrum? It's what you guys are doing right now.
Thinking.
Oh, there you guys go. So this is what? Higher mental function. So all your higher mental function, if you guys notice, there's a big chunk of it, right? That's dedicated to that. So higher mental function equals cerebrum. Excellent. Okay. We have this white portion. You guys see this is white, right? Don't tell me it's like ivory or something like that. Okay. It's white. Okay. This white portion that you're seeing in the middle is in general called something. What is it called in general? It's called the diencephalon. Okay. And the diencephalon is comprised of three parts. Okay. And we know these. I told you. I think we went over these together. That little egg-shaped structure, kind of in the middle. What is that thing called? Thalamus. Up here, the white. Okay, we have the thalamus. Okay, we have the structure that's located below it, right, which is the hypothalamus, and the structure that's behind it, back here, which is called the epithalamus. Okay, so these three structures together, we call them the diencephalon. Okay, the function of the diencephalon in general is sensory integration, sensory input. Okay, it processes sensory information. Does that make sense? Okay. If we look at number three, what's number three? Which is the, let me clear this out. If I clear it out, it's gonna get messed up, but let's try to clear it a little bit. Okay, do you guys see that little purple area, kind of like in the middle there? Is it purple?
Yes.
It's divided into sections, yeah. So it starts with the purple, which is the midbrain, then that light kind of teal color, the pons, and that darker blue color, medulla oblongata, okay? So you guys got to know this, okay? What does the brainstem do? What does the brainstem do? It coordinates visceral activities. What does that mean? Coordinates visceral activities. I told you guys this. One of the
Balance and movement.
Organ.
not balance and movement.
Heart rate.
Breathing, heart rate, because these are viscera that control these things. Visceral movements, right? Involuntary movements. Vital, right? Very good. And the last structure there is number three. I think I skipped it earlier, but number three is this green section here, right? Looks like a little cauliflower. This area right here. That's called the cerebellum. And what's the function of the cerebellum? Balance, coordination, those kind of things. It coordinates muscular activity. Balance is one of those things. Okay? You guys cool with that, just in general? Okay? I'm gonna, of course, go deeper into it. This is just a snapshot, right? Just a quick little summary, okay? Just to get you guys comfortable, okay? So brain, spinal cord, we're gonna talk a little bit about embryonic development, and then we're gonna go back to all these structures and functions. Make sense? So there are a couple questions that talk about embryonic development. I'll kind of highlight those for a bit, or in a bit. But I want you guys to understand one thing first. Really important. When I say really important, what does that mean?
It's going to be on the quiz.
That's a quiz question, so write it down. Where does the brain, if we're talking about embryology, right? Embryo. Where does the brain and spinal cord develop from or arise from? There's a structure called the neural tube. That's what you see in picture A, okay? That's the neural tube. The anterior or rostral portion becomes what? The brain. The posterior or caudal portion becomes the spinal cord. So let's go home early. When I pause, I'm not thinking, I know, I want you guys to complete. So posterior,
Cerebellum.
Spinal cord.
spinal cord. Spinal cord. Anterior, brain, right? Anterior brain. So where does the nervous system develop from embryologically?
Neural
The neural tube, right? The neural. You guys are throwing me off now. The neural tube, okay? The neural tube, okay? Very important that you guys remember that, okay? So really know that. If we're talking about embryo, everything arises from what? The neural tube, okay? And we can subdivide the brain into a couple sections, okay? We divide it into what is called the forebrain, the midbrain, and the hindbrain. You guys see that? Forebrain, midbrain, and hindbrain, okay? And like I said, the posterior part becomes the spinal cord, okay? The openings, they become the ventricles. What's the significance of the ventricles? They're kind of like these hollowed out structures that we'll look at later. They're gonna allow for what to circulate through them?
CSF.
CSF. Is there blood in the brain? No. It's a definite no. Because, talking about, so Eddie brought it up. So why don't we have blood in the brain? So this goes back to a lecture point, which is going to be an exam question.
Because of the blood-brain barrier.
Perfect. And what cells?
Astrocytes.
See? I think you just said it just so you sound smart. Okay? And you get a quiz question for the class. That's exactly it. That's exactly right. The astrocytes form the blood-brain barrier. Quiz question for Saturday? Most definitely, right? Now you guys care about it, right? Whatever. Okay, so you guys understand this dynamic so far? If we talk about secondary brain vesicles, right? We're going to have essentially 5 structures here. The forebrain becomes the telencephalon and the diencephalon. The midbrain basically is unchanged. It's going to be undivided as the mesencephalon. And the hindbrain, if you guys notice, becomes the metencephalon and the myelencephalon. So I know these are a lot of like crazy terms. You don't have to know these terms. Did I say you have to know them? Did I say they're super important?
No.
No. So right now you're just listening, right? Okay. This is really important, okay? So D, you gotta know, right? You gotta know the adult structures, okay? So you gotta know the adult brain structures. They're really important that you know, the cerebrum, the diencephalon, the brainstem, midbrain pons, medulla, cerebellum. Those are important structures to know. But should we kind of just get an understanding of where they're arising from? Do you guys see the kind of evolution of the brain as, the embryo develops into a fetus, right? So again, really important to know the adult brain regions that you're seeing here. They're color-coded. You got to know that. That's really important. And you got to know that everything begins as the neural tube. Okay, that's the most important thing to know. Here again is another view of embryonic development. Everything's color-coded for you guys so you can actually see it. I think it's just interesting to note how the hemispheres kind of grow, if you notice, into this kind of horseshoe shape. Do you guys see that? Basically, it kind of grows into this horseshoe shape. And it continues to grow. And as it grows, do you guys see more folds kind of take over, right? More folds, more wrinkles, more creases. So again, at birth, just about at birth that you see here, you can see a very similar pattern of convolutions of the structure as an adult, right? As an adult brain would have. You can see all the structures have been formed already. So again, should we be able to identify these structures readily, right? Most definitely we should be able to, okay? So maybe we should correlate just a couple of them real quick. Maybe just note a few, just for fun, okay? Like the telencephalon, right, becomes what? It's this, the cerebral hemispheres. The diencephalon, can you guys break that down for me? What's the diencephalon? Thalamus? Hypothalamus, epithalamus, very good. The mesencephalon, M for M, is the midbrain, very good. The metencephalon would be the other 2 structures would be the pons and the cerebellum, okay? And the myelencephalon is the last part, MY. Y is pretty far down the alphabet, so that would be what? The medulla oblongata, okay? Okay, so these, again, you gotta know these ones for sure, okay? So let's kind of understand function when we look at these adult brain structures. If we're talking about, the divisions of the brain, we can divide it into the cerebrum. The cerebrum is the largest portion, true or false? True. Does it process sensory input? No. Does it deal with the coordinated movement of muscular activity?
No.
No. Does it deal with higher mental function?
Yes.
Perfect, okay. The diencephalon, the purple that you're seeing there, does that process sensory input?
Yes.
For sure, okay. The cerebellum, does that deal with the regulation of visceral activities?
No.
No. Does it deal with the coordination of muscular activity or movement? Yes. The brainstem, does it coordinate and regulate visceral activity?
Yes.
Yes. Okay. Should you know the functions of each, like just in general? That's not that hard, right? Okay. Here it is kind of in isolation, right? So here's the brain that we've been seeing so far. Here's our key that we can review, and then here's just a closer view of the different structures. So again, if we look at the diencephalon, we know it's comprised of the thalamus, of the hypothalamus, that's right below it. We don't see the epithalamus. Epithalamus would be back here, right? We'll talk about the epithalamus later. The brainstem is made up of three parts, which are what? Midbrain, which is first up here. Then you have the pons in the middle, and then the medulla oblongata. So again, should we know where these structures are found just in general, right? For sure. Are we understanding a bit more about their function?
Yes.
We're getting it, right? We're understanding it a little bit, right? Hopefully. Okay. Remind me, the brainstem, what does it do?
Balance, coordinates the visceral function.
Visceral activities. Cerebellum,
Balance,
what does it do?
coordinate muscular
Coordinates muscular activity. Diencephalon,
Sensory.
sensory input. Cerebrum,
Higher mental function.
thinking, right? Processing. Yeah, higher mental function. So if you're not able to come up with the answer like this quick, that means you've fallen asleep. Okay? So we've already repeated this information how many times so far? When I say we, I mean me. Well, you guys too, you guys have helped out. We've already said this about like 7 times so far. So we should know it by now, right? 10 times it's in our, we can't forget it already, right? So we should know this stuff pretty straightforwardly, okay? If we look specifically at these structures, again, just in general, the midbrain is going to control some reflexes. The pons kind of deals with breathing, more or less. And the medulla oblongata controls, like we said, heart rate, blood pressure, other reflexes like coughing, swallowing. Those are all kind of under the charge of the medulla. Oh, there they are, okay. Okay, spinal cord, the only thing that we'll kind of mention about the spinal cord here is that it's going to exit the skull at what level or what structure? The foramen magnum. You can see this on CT, MRI right here, mid-sagittal cut. You can see the brain. You guys see the cerebrum?
Yes.
Yes.
If I show you an MRI, I mean, not anymore, but if you were rotating with me and I asked you, what do you guys see here? Can you guys find the cerebrum, or do you guys see anything abnormal or weird? Again, as a clinician, I'm asking your input. You should be able to say, well, yeah, there's a clot there. It looks good there. There's some, something that's going on there. You should be able to tell me general anatomy, right? No quiz question, but just understand the MRI looks almost like the image, no? You can see this area here. What is this area down here?
Cerebellum.
Cerebellum. This is what structure in the lab that we saw. We haven't seen it yet, but we saw it in the lab. The corpus callosum that unites both of the structures. And then you can actually see this other structure here, part of the brainstem, the pons. That means that this area right here would be what? Medulla. And the area up here? Midbrain. Yeah, perfect. You guys just read an MRI. That's hardcore. Okay? Nice. Good job. Region to the brain, the way the brain's organized, we know that, if we look in general at the general aspect of the brain, pretty much similar in pattern, left and right. There's additional areas if we do like a cross-section of the brain. If you guys notice, the outer portion here, do you guys see the gray? Right? That's called what type of matter?
Great.
Gray matter. What does that mean?
Non-myelinated.
Not myelinated or non-myelinated, very good. And you guys notice there's white. You guys see the white matter here? Myelinated. But you notice that within the white, there's also regions, I'm going to kind of color them in blue right now, like here and here, that are in what color? Gray. So you have what are called nuclei, right? And these nuclei have very specific functions. And there's a ton of nuclei in the brain because does the brain do a lot of things? Yeah, so there's a lot of nuclei within the brain that have very specific functions. Okay, we're going to look at some of them, not all of them. Okay, but just understand the general features of the brain. Does that make sense? There's white matter, there's gray matter. I know what the cortex is. What's the cortex? The outermost part of the brain, right? The cortex is the outermost part. What type of matter is it comprised of? Gray matter. It's myelinated. So here, Andrew, this here is the cortex. That's the cortex, okay? Are there gray areas within the brain itself? For sure. What are these openings in the brain? They look kind of like cavities. What are those? I mentioned it earlier. This is what allows CSF to circulate. Ventricles, right? Those are the ventricles of the brain, okay? So if you were to do like a transverse cut of the brain, like this, okay? And you lift the brain up, you're gonna see these like holes in there, right? Those are called ventricles. What's the purpose of the ventricles? To allow for CSF to do what? To circulate, okay? All right, good. Okay, when we look at the brain itself, we know that it's going to be encased in something, right? What is it encased in? Skull, okay. What part of the skull? The cranium, okay, good. Okay, the cranium, okay. Cranial bones, right? So if we remove the cranial bones, we can kind of see the brain itself. This is where it's housed. But there's also, remember, like we said, with every single organ that we have what? A covering, right? So there's a covering to the brain as well, just like the heart. We said it's the pericardium, the lungs are the pleura, the intestines is going to be the peritoneum. The brain has its own covering as well, called the meninges, right? The meninges, yes. And there's actually like 3 parts to the meninges, okay? So we got to know the meninges. It's important to know, okay? We know the bones. I'm not going to review the bones because we've seen them before. The meninges are new to us. You can kind of see the way they're kind of broken out here. The dura mater, that's the outermost layer, right? It's going to be comprised of dense, fibrous connective tissue. You have the arachnoid matter, which is this kind of very thin, transparent membrane, and the pia mater, which is the innermost and most delicate of the coverings. Now, the significance of the meninges, the covering of the brain, right? And when we do the sheep's brain on Monday, right? You're going to notice that you're going to have to take off the meninges, by the way, right? So you're going to have to, document that. The meninges. What's the clinical significance of the meninges? There can be sometimes a disease, right? Of the meninges called meningitis, right? Yes. Common site of infection, common site of intracranial bleeding are the meninges, right? So let's take a look here. You don't have to know all this stuff here. This is, pretty advanced. This is the pathogenic cause of meningitis by age group, right? Because it's interesting to note that depending on your age group, I highlighted the blue is the most common in an age group population, right? So you test for those types of microorganisms depending on your patient's age. But the thing I want you guys just to remember about meningitis is, what is it? It's inflammation of the meninges. It's inflammation that's caused usually by some type of infection, yeah? And is this kind of like a thing that's like a cold, like, oh, no big deal, you're fine? No, it's very serious, right? It's very, very serious. It is life-threatening. It can cause encephalitis. That's what you see here. This is hardcore inflammation of the brain, of the parenchyma, essentially, that can cause death in some cases, right? And if not death, sometimes you have severe sequela from that. Sometimes the person has paralysis, right, after that, because the brain what? Swells up, basically. And if you guys remember your cranium, is there space for the brain to move? There's no, it's custom built for the brain, and it really can't move much. So anytime you deal with inflammation, that can cause an issue to the internal function and structure of the brain, right? So it's very serious. How do we make the diagnosis of meningitis? What do we have to look at? The CSF, yes. We have to do basically a lumbar, right, extraction of CSF. And what color is CSF?
Clear.
Why?
No blood.
No blood. Why? Blood-brain barrier. What provides that? Astrocytes. You guys are good. So you guys are awake. You guys were scared me for a second. My confidence in you is restored. Okay, good. You guys see, like, you see what I'm saying? Like, if you guys know it, you're able to. We can have a conversation now. Yeah, if we're talking about meningitis, I can ask you all these questions. Is that fair? If you're a clinician, if you're a newbie, if you're coming in, for sure. Yeah, because I want to know what you know. Do you know about the nervous system? If I show you what's the point of me showing you a CT scan if you don't know the anatomy of the brain? What am I wasting my time for? No? If you know it, All right, now we're talking, okay? So just be aware of that. But you don't have to know any of that stuff aside from just the inflammation aspect of it. Okay, ventricles. How many ventricles do we have? 4. What type of cells line the ventricles? Which are these types of cells? They have little cilia on them. Exam question, right? Or Saturday, right? Ependymal cells, right? Ependymal cells, they line the ventricles, and their main function is what? To produce CSF, right? Do you guys see the blue here? These are the ventricles. They kind of highlight them in blue. You guys see that? So when we look at them in real life, they kind of look like this, like these kind of hollowed out structures. You guys cool with that? You guys understand that? Those are the ventricles. So the main function of the ventricle is what? Allowing for CSF circulation. How many ventricles are there? 4. Now on the lectures, I talk about each one. I'm not going to ask you any questions about the ventricles because they're pretty crazy, right? You can listen to it, which is good to understand, but I'm not going to ask you any questions about specific ventricles or locations of. Oh, do I ask you about locations? Should I ask you about locations? No? That was a fast no. Too fast. I don't like that no. Isn't there one on the? I think they do ask you one on the brain. The model. Did you guys locate the ventricle? I think it was the 4th ventricle. You guys find that one? No? Did you guys look at the brain model? Yeah. Was there a lateral ventricle or the 4th ventricle?
It's a
Lateral ventricle. So the one I want you guys to keep in mind, just keep this one in mind, is going to be the lateral ventricle. Okay, the lateral ventricle basically are the little. I think I highlighted them here for you guys too. Do you guys see these ventricles here? They look like C's, like little. From the front view, they look like little horns of a ram or something like that. But those are the lateral ventricles. You guys see them here? Lateral ventricles. Those are the ones I just want you guys to keep in mind. So if I ask you a question or on the lab or on the lecture that talks about ependymal cells and CSF and a lateral ventricle, you guys understand what I'm talking about, right? You guys know what that means, right? You guys see the lateral ventricle here, right? Okay. That's an important one just to keep in mind. That's it, okay? So what's the purpose of the lateral ventricle? Perfect. What kind of cells line it? Ependymal cells, very good. There's a special type of capillary actually, you're seeing that here. Yeah, we have the ependymal cells. There's actually a special type of capillary called the choroid plexus. I think that's another one on the key as well. I think we were looking at that one, the choroid plexus. So note that one as well. The choroid plexus is also good to know just because it's a specialized capillary network that extends from the pia mater that secretes the CSF, right? So this dynamic of ependymal cells and the choroid plexus, they work together to do what? To secrete CSF. So just make a note of that right there. The choroid plexus, a specialized capillary network. They don't secrete blood, right? That's the thing. They arise from the pia mater, which is the innermost covering of the brain, right? And what do? They help secrete CSF. That's the most important thing to kind of keep in mind. And the majority of my CSF is produced from what ventricle, by the way? The lateral ventricle, okay? So that's maybe important. Should I keep talking? Sure. What's the purpose of CSF? What's it do? Protection, yeah. One of the main was protection. If CSF wasn't in your skull, your brain. Have you guys ever touched the brain? Ever of any animal or human or anything like that? No? How are brains? They're super delicate, right? Super delicate. Yeah, they basically are, it's almost like Jell-O basically. So they're really, really delicate. If your brain was just kind of like moving around in nothing, you'd basically splatter your brain pretty quick, right? So you have to have something that's going to immerse your brain to protect it. So protection is a big thing, right? Protection, that's one. What else is found in CSF? When we do a spinal tap, you guys said earlier, right? I know we're looking for microbes and stuff like that, but what else is in the CSF? And that'll tell me if it's bacterial, if it's viral, if it's parasitic, if it's fungal, in terms of my diagnosis for the microorganism. Well, proteins, right? Protein content, so protein is found in CSF. What else is found in CSF? What's my brain use as an energy source? Athena knows. Glucose, right? right? Glucose, sugar, right? So am I going to find glucose or sugar, or not sugar, glucose in CSF? Yes or no? Yes. Okay. So does it provide nutrients? Is that one of the functions of CSF? Yeah. Okay. So do we understand what CSF does? Okay. Protection is number one, and then nutrient content is number two. Okay. That's it. Is that okay with you guys? Good? Comfortable? Done? Again, I'll highlight more here. You can hear the audio. I'm going to kind of skip over a couple. So what's the one that produces the most, the majority of the CSF? Lateral ventricle, right? Do you guys see the lateral ventricle here?
Yes.
And then here? You guys cool with that? Okay, and then here's basically the flow of CSF. There's like two pictures here. Again, I'm not going to ask you any questions specifically about the flow, but just understand that the ventricles, right, the way they're designed, it allows for that CSF to kind of flow. And does that CSF flow through the spinal cord as well?
Yes.
Yeah, because when we do a spinal tap, right, what are we pulling? Fluid from where? The CSF, the spine, right, the spinal cord itself, right? So yeah, it's going to encase or encapsulate the entire central nervous system, right? I know we're focusing specifically on the brain, but this fluid encompasses or encapsulates everything. And there's a flow to it, right? Right? It kind of flows, it's recirculated. Because we need what? Two things. Very important that CSF does. One, protection. Two, nutrients. Done. Perfect. What type of nutrients?
Glucose.
Good. What else do we have in CSF? Proteins, right? Proteins are really important for diagnostic purposes, like I said before, to figure out if it's viral, bacterial, fungal, all that kind of stuff. We're going to stain it too, of course, but the protein content is also very important to know. So again, it just gives you a breakdown of fluid dynamics in CSF. Cerebrum. Okay, cerebrum is the largest part of our brain. You know, it controls all our higher functions. So does it control things like thinking?
Yes.
Yeah. Does it control our memory?
Yes.
So am I testing you guys every couple seconds by asking you a question again? Because what am I testing? To see if you're remembering and being able to recall what we're reviewing. Yeah? So I'm testing your cerebrum, okay? Your cerebral function. Does it help regulate my speaking, thinking, understanding of things?
Yes.
Basically, what do we kind of encompass this as? Higher mental function. Does that make sense? It's kind of like an all-encompassing thing. Okay. So, we have to understand that the cerebrum, we can break it down into what? Hemispheres. You guys, like in the lab, you guys remember we took the brain apart, we had a left and we had a right hemisphere, correct? So we're going to understand the hemispheres. We also have to understand the surface anatomy of the cerebrum, because if you guys notice, there's a lot of convolutions on the cerebrum, right? You guys notice that? Things are popping up, things are popping down, right? So we got to figure out what those things are called, okay? This is called surface anatomy. We're going to look at the surface anatomy of these structures. So the first thing we have to understand are the lobes, right? So how many lobes do I have here? We have 5 lobes. We have the lobes that are going to underlie the bones, right? So we have the bones on the superficial part and the brain that's going to be deep to those bones. So do we have a frontal lobe?
Yes.
Can we identify that frontal lobe? Do we have a temporal lobe? Do we have a parietal lobe, an occipital lobe? Okay. What am I missing here? There's one lobe that's missing, right? That we can't see. If you guys notice in this picture down here, you actually have to move the frontal lobe a bit, and you have to move the temporal lobe a bit, right? By these little separators, and you get to a deeper lobe. That's called the insular lobe. It's insulated, right? So that last lobe is when you can't see superficially. That's called the insular lobe, okay? So do the lobes make sense to you guys? So we have to know the lobes, sulci, fissures of the cerebral hemispheres, okay? We have to know this stuff. So we got to get comfortable with each one of them. So we're kind of comfortable with lobes? Easy, right? Lobes are easy. Okay. Let's go to the next thing. Sulci. Okay, so sulci, we got to understand that we have ridges on on the cerebrum, on the surface. We can talk a little bit about surface markings. So these little shallow ridges, right? These little bumps that you see here, those are called gyri. Okay, you have gyri that are little bumps, right? Like a little speed bump, right? That's a gyri. Okay. Let me highlight that for you guys. Okay, and then you guys see that little depression, a little depression that you see there, a little depression that you see there, a little groove, right? That's called a sulci or sulcus. Okay, sulci or sulcus. So you guys see it? A little depression, a little groove. Okay. If it's really, really deep, right? What do we call it? A fissure. So anything like below that little line there, that all that stuff down here, that'd be a fissure. So deep groove is a fissure. Does that make sense?
Sure.
Good? So we got to know the anatomy here, okay? Just in general. The sulci are kind of important because it kind of helps separate the brain into lobes. So we're going to understand these kind of lobes and sulci as we kind of advance. But for now, it's just important to know just general stuff. Fissures. Okay, fissures you got to know. Okay, there's a lot of fissures to kind of be aware of. Let's start at the top and work our way down. We have the first one here. We have the longitudinal fissure. So what is that longitudinal fissure doing? It's separating the two hemispheres, right? Right from left, or left from right, whatever. Okay, so that's a big one. That's a longitudinal fissure. We got to know that one there. We have what's called a transverse fissure. Do you guys see the transverse fissure? Basically, it's going to separate. It's going to be this one here. I'll draw it right now. I think it's there somewhere. There it is. Transverse fissure. It's going to separate the cerebrum from the cerebellum. You guys see that? So there's a transverse fissure that separates the cerebellum from the cerebrum. Are we ever going to confuse cerebellum with cerebrum? No. Okay. Cerebellum is going to coordinate muscular activity. Cerebrum controls higher mental function. We know where that's located. Okay. We have the central sulcus. You guys see this like V right here? You guys see that one? That's called the central sulcus. And it divides something into something. What's it divide lobes into? Frontal from? Parietal. Very important that we know that. Frontal from parietal. So what sulci separates the frontal from the, what was the other one? Parietal. Central. Transverse?
No.
Longitudinal?
No.
Lateral? No. Okay. Central, right?
Central.
Okay. The lateral, why is that one important? Because that separates temporal from parietal. You guys see this one here on this side? That's the lateral. Parieto occipital. I mean, the names tell you, right? Basically, if we said parieto occipital, what does that separate? Occipital and parietal. And the transverse cerebral fissure separates the cerebral hemisphere from the cerebellum. We already talked about that one. Oh, same one. That's repeated twice. It's just transverse fissure. We already said it. Are we comfortable with the sulci? We good with those? Make sense? So of these, the most important one, again, if I was a student, right? If I got to know my information, I'm going to organize it into what's most important, right? So what's most important here? Which fissure do you think is the most important one? Probably two, right? Probably the transverse, the central and the longitudinal. Those are pretty important, right?